Just want to thank the ARM committee for our presentation here from Avectas. So I'm going to talk to you today about the manufacture of healthy and highly functioning gene-edited immune cells from Avectas. So, what we've heard throughout this morning and this afternoon, there's multiple challenges that exist with the editing of primary immune cells. And for example, we see in the previous presentation the need to multiple or multiplex edits. And so when you perform those multiple edits, that can require additional manufacturing time or duration to allow the cells to recover before you do the next edit. So really one of the challenges is to decrease the manufacturing time to come up with a more efficient process. Additionally, as was noted previously, is the ability that cell health can be negatively impacted by the delivery technology or the cargo that you're using to do that edit. So the cells can be unintentionally differentiated or exhausted due to the gene editing step. And that can result in a cell health that is not as good as it's intended to be. Ultimately, what that can result in is a cell that is not as functioning as you'd like it to function. So the decreased cell function post-editing can potentially reduce the tumor killing capability in oncology applications and ultimately negatively impact the clinical efficacy. So it's definitely there's multiple challenges exist with the current gene editing technology. What we've developed at Avectas is the Solipore system. This is an easy-to-use, automated, closed system that's ready for evaluation. It's highly automated and is able to do reproducible transfection. And what we've been able to show is the ability to do a broad range of different types of gene editing cargoes, messenger RNA, plasmid DNA, RNPs, the CRISPR tools that we're able to utilize and deliver effectively with the Solipore technology. We launched this earlier this year, and we'll be GMP ready by the end of 2023, the end of this year, to utilize this technology in a cell manufacturing process. We're also able to work with partners under co-development and licensing agreements to evaluate this technology as well. So a little bit more, what is Solipore all about? How does it compare to existing technology? It exists of three components. The first, which you see, is the multi-use controller, which consists of both the hardware and the software that make up Solipore that actually controls it, the mechanics, the pumps, et cetera, that are utilized to control the technology. The second piece, we have a proprietary reagent solution that's used at the top to formulate different types of cargoes that's used to deliver the formulated cargo and do the transfection. The third piece, which I'm going to get to in more detail, is a closed single-use transfection chamber where the transfection actually occurs. So these three components make up the Solipore system and utilized to produce cells, as you see in a couple slides, that have superior cell health and function relative to existing technology that exists. So a little bit more of how actually the Solipore process works. It actually is able to produce in a very rapid manner under physiological conditions, a very healthy and highly functioning cell. So the first part of the Solipore process, cells that need to be edited are introduced into a closed single-use transfection chamber in any type of cell culture media that's desired, again, under physiological conditions, so no need to concentrate the cells. The cell culture media is removed. These cells sit on top of a membrane filter, and at which point we use a precision atomizer to spray or deliver the cargo that's formulated in a proprietary solution. That solution temporarily permeabilizes the cell membrane and allows the cargo to enter the cell, at which point the editing occurs. After the editing step, the cell culture media, it can be any media of choice, is reintroduced in the chamber, and then the cells are then removed from the chamber back into a bag where they can be further processed as part of the manufacturing process. This whole process takes about 10 minutes process time, so it's quite a rapid step to do in a manufacturing process. So I did want to go over, with that introduction of the Solipore technology, a little bit more about some of the data that we've been generating that really illustrates we're able to generate a healthy and highly functioning gene-edited immune cell. So you'll see some of the data with respect to the delivery of very relevant functional cargo to primary immune cells. We've also done some studies in which we've done multiple or multiplex editing of T cells and done a good amount of in vitro cell characterization And also with some in vivo, we did an in vivo CAR T cell efficacy study. It really illustrates the advantage of Solipor relative to the existing technology. So if you look at some of the data, and this is a dose response, looking at several different targets, TRAC or TCR, CD7, as well as beta-2M in the form of an RNP as the gene editing tool. In this particular case, we see that we achieve efficient transfection efficiency, and we're able to achieve levels by increasing the amount of cargo that is delivered, or we call it sprayed, to the cells. We can deliver transfection efficiencies 60 to 90%, and it certainly varies with the type of cargo you're delivering, but we're able to achieve quite high levels of transfection efficiency with these different types of edits that we're performing. So this really illustrates Solipor can deliver functional cargo to primary T cells. If you look at what we're able to do with respect to how the cell functions, we've done a good amount of in vitro cell characterization utilizing T cells that have actually gone through a triple edit knockout of those targets that I just went over, and we looked at the overall cell health after they were edited. Some of the results that we look at with the multiplex editing of T cells include both cell health as well as phenotype. And so you can see, looking at cell apoptosis, the graph here you see on the left really looks at early and late apoptosis, and you can see relative to electroporation, in which you have significant apoptosis occurs, the level within Solipor is significantly less than that, and favors quite comparable to that of an untreated or a control experiment that was done here on the lower left. So you can see we produce a cell that's edited that is less apoptotic. Similarly, looking at stem cell memory population, comparing Solipor to electroporation as well as against a control, the graph here on the right shows that you're able to retain a large amount of stem cell-like memory population, which again in turn demonstrates the ability of Solipor. It's a very gentle process, a rapid one, that results in a cell that retains a high level of cell health. So as we look at this, we also want to do some additional in vitro characterization to understand, will this ultimately translate to something that has potentially higher tumor killing capability or clinical efficacy? So we did some additional in vitro characterization using cell avidity. And again, this is becoming more and more showing a correlation between the ability to have a high avidity level and the ability to have in vivo efficacy. This looks at the overall binding strength amongst the different interactions between the target as well as your CAR T cell in this particular instance. So we did some avidity studies and compared the Solipor technology to electroporation. And you can see shown here, and this consisted of T cells that were transfected with those 3 knockouts I mentioned before, following a transfection step that was done both on electroporation as well as Solipor, as well as a control in which no transfection occurred. We followed that by a lentiviral vector transduction step, and then we measured the cell avidity. So it really measured the effect of the transfection on the cell avidity. And you can see when comparing a control sample that's in gray compared with Solipor and electroporation, the Solipor is able to achieve higher levels of avidity compared to electroporation and comparable levels to a control study. So it really shows, in this particular case, as a CD19 target, the ability of Solipor to produce a more highly functioning cell and one that has a potential of a higher tumor killing capability. So what effect does it have on the cell function? When you look at taking Solipor cells after the editing occurs, and then you re-stimulate those cells, and you look at the inflammatory cytokine response with some different markers, you can see with different types of markers and cytokines, interferon gamma, granzyme A, granzyme B, as well as granulysin, you see a higher amount of these markers or cytokines produced, which in turn illustrates that following the Solipor process compared to electroporation, we get a higher level of in vitro cell functionality that occurs. So overall, in turn, this shows with the in vitro work, Solipor is able to produce a more healthy and highly functioning cell. Now, ultimately, how does this translate to the performance in an in vivo study? And so we did do some additional work in which we took Solipor and did delivery. In this particular case, we did an mRNA CD19 CAR, and with the xenograft mouse model, we looked at the ability of Solipor edited cells, compared that to electroporated cells, and how they could effectively kill tumors that are engrafted in mice. And so this is a mouse model. And what you can see shown here with the figure on the left with an appropriate control in which the tumor and the mice continue to grow with the blue color here. When we compared cells that were edited using electroporation compared to that of Solipor, the figure in the middle illustrates that the level of tumor was greatest in the electroporated cells compared to the cells that were transfected using the Solipor system. And in turn, we had a higher level of engrafted CD8 cytotoxic cells shown here with the graph on the right. So overall, this really illustrates the fact that we have a more healthy and highly functioning cell in vitro leads to the ability to have a more a potential for higher tumor killing capability when translated to an animal model. So overall, this really illustrates the potential of this technology to generate a more highly and healthy cell that can result in a better product at the end. So just to recapitulate some of the key areas in which Solipor provides some advantages is the ability to perform complex or multiple gene edits that results in improved cell health and function. That's going to really be needed for the next generation of cell therapy manufacturing. We can see that we're able to maintain improved cell health and phenotype. We saw some data looking at the improved stem cell-like memory and lower apoptosis, leading to ultimately improved proliferative capacity, improved engraftment of the cells. And as a result, we had a T cell that were highly functioning in both in vitro as well as in vivo studies. And since we recently launched this technology, it's highly automated, easy to use with minimal cell processing, and provides a lot of flexibility for the developer and manufacturer to adjust to optimize their manufacturing process with the use of this technology. So just want to say right now is a really unique opportunity to partner. We're here today to talk to people that are interested in this technology. And as I mentioned, we'll be ready for GMP at the end of this year. And we feel it has really differentiated technology relative to what's currently existing on the market. So I want to thank everyone for your attention.